On behalf of the New Technology Committee, I would like to thank APSA and welcome everyone to the technology session. And thank you for joining us wherever you are. There will be 4 presentations in this session. 2 will be the innovative abstracts and 2 didactic presentations. Each abstract will present and we'll have 2 invited discussants, and the didactic presentations will have live questions and answer via a chat feed. Also, I wanted to remind those who are invited as voting members to vote not only these 2 abstracts, but all the abstracts in innovative sessions. And an award will be given to these researchers for their wonderful and creative work. The first abstract will be presented by Dr. Robert Crum from Boston Children's Hospital. The title is The Eye Fastener, a Novel Tissue Apposition Device.
Thank you for the opportunity to present our work on the eye fastener. Authors of this presentation are co-inventors on a provisional patent submitted for this device. Tissue apposition is an essential element of nearly all surgical procedures. With minimally invasive techniques, tissue apposition can be particularly challenging, such as in the gastropexy step of pediatric laparoscopic gastrostomy tube placement. Devices and techniques have been developed to address this challenge, but are not ideal for the pediatric population. One such device is the T-fastener. While effective, it consists of a non-absorbable metal anchor and an external suture lock. These components are associated with migration, infection, skin erosion, pain, and reoperation. One commonly used technique is the transabdominal suture, which can be cumbersome and is especially difficult in patients with thick abdominal walls. To address this problem, we sought to create a device that opposes 2 tissues using minimally invasive principles, has no external components, is made from bioabsorbable material, and can serve our pediatric population. Working within the Boston Children's Hospital Surgical Innovation Fellowship, we vetted our idea with a group of multidisciplinary advisors and broadened the need to include the possibility of opposing any 2 tissues or any tissue to implant. Initial designs were developed using SolidWorks, and prototypes were printed with an Eden 3D printer. We call the device the eye fastener, and it has 2 major components. The first is a single-patient, multi-use, spring-loaded ratcheting plunger. The second is a needle housing 2 bioabsorbable anchors along a single bioabsorbable suture. These anchors within the needle each have 2 holes through which the suture passes to allow for free movement of the anchors along the suture. To deploy the eye fastener, the needle is first loaded onto the plunger. The operator then passes the needle tip into the desired location, depresses the plunger once to deploy the first anchor, then withdraws the device to the second desired location, depresses the plunger again to deploy the second anchor. The needle is then removed, and the 2 suture ends are tied together, opposing the tissues between them. Here we show this final step in further detail. Once the anchors are deployed into the correct location, the suture is pulled tight and a knot is formed. As the knot is tied down, the anchors are cinched together, securely opposing the tissues between them. A second method for apposition is shown here. The barb suture acts on itself in a zip tie type mechanism, whereas the operator pulls the suture until the anchor engages the fascia. Continuing to pull up on the suture brings the anchors together, creating a secure apposition. Our latest prototype is demonstrated in this ex vivo model of gastropexy. The first needle is loaded onto the plunger. The needle passes through all layers of the abdominal wall and into the stomach. The plunger is depressed and the first anchor is deployed. The needle is withdrawn to the subcutaneous space, and the plunger is depressed again to deploy the second anchor. The needle is removed from the patient and set aside. The free suture ends are tied together and the knot buried to oppose the 2 tissues. The suture is cut at the skin, and the tails retract into the subcutaneous space. The second needle is loaded onto the same plunger. Here we can see deployment from the operator's point of view. We have found that angling the device helps to deploy the second anchor when the subcutaneous space is small. Cross-sectional view demonstrates a secure gastropexy ready for your gastrostomy tube. Our next steps include investigation of ideal bioabsorbable materials and comprehensive structural testing. From there, we will pursue in vivo testing and FDA approval. We believe our device can be used for many procedures, including the pediatric gastropexy demonstrated here and laparoscopic ventral hernia repair. The iFastener device allows for easy and secure apposition of two tissues through a single needle hole with no external components. It is applicable to a wide range of procedures and populations, including pediatrics. Thank you.
Thanks, Dr. Nguyen, for inviting me to discuss this abstract. I would like to first congratulate Dr. Crum and team for their innovative work and excellent graphic presentation of this device. My first couple of questions are really just technical questions for Dr. Crum. How many eye fasteners do you anticipate will be needed for a standard lap G-tube in a pediatric patient? And secondly, do you think will each eye fastener require a separate incision, or would it be possible to deploy the eye fastener through the primary G-tube incision site?
Thanks so much for that question, Dr. Johnson. Typically it depends on the technique you want to use. We anticipate being able to use 3 to 4 fasteners for your traditional laparoscopic pediatric gastrostomy tube placement. And regarding the number of incisions, the way we've described it here is through a separate skin nick, just a needle hole size incision. There has been some research publishing a technique in which T-fasteners can be placed through the gastrostomy hole and then tied to themselves. So that remains a possibility of the eye fastener. However, it's important just to keep the implants a little bit, to provide a little bit of distance between your gastrostomy tube and your fastener.
Secondly, you mentioned that this is a reusable device. How would using this device in place of other current methods of gastropexy affect the overall cost of the procedure?
Yeah, the price point for this device is not yet determined because we have to do a lot of testing to find out what materials we will be using. The needles themselves will be single use and disposable, and then the plunger we anticipate will be single patient, so you could use it with multiple needles, but the plunger would be used for an entire procedure and then disposed of at the end. We think this is applicable just because the components that would go into the plunger are relatively inexpensive to produce. We anticipate the price point to be above that of a standard T-fastener. However, the savings would need to be proven with a clinical trial.
And finally, so once your device is approved and ready for clinical use, how will you convince your colleagues to adopt this technique?
I hope we get to that point. We intend to primarily use our position as physicians and surgeons, and we think that's one of the important things we do within our fellowship. Coming to a meeting every couple of years and providing updates on the technology and really getting down to a clinical trial to provide some solid evidence that this really is a better solution for gastropexy and other procedures.
Dr. Crum, this is Samir Pandya. Thanks so much for your work. I found that it's very innovative and I applaud your efforts. I did have a couple of questions for you if that's okay. What my concern is that when you tie down your stitch, you now will have two little, shall we say, barbs. The T-fasteners have barbs, but you're proposing some bioabsorbable material. Those two little barbs will be squeezed down tight with tissue being apposed in between. And the smaller that cross-sectional area is, the higher the chance of that eroding through.
Yes, that is a legitimate concern. At this point, we look towards T-fasteners and their dimensions, and we see that the current ones are about 1 centimeter long, and they fit within a 20 gauge needle. That's the rough surface area that they have. So we're really talking about bringing those two together. There are reports of migration of T-fasteners once they are placed, probably due to the pressure. But we think that that type of surface area will be reasonable once we establish a material to be used for it.
I often use the T fasteners as just a pure retraction type of device when I do single site laparoscopy or something like that. And so what I find that's useful in that is that when I'm done with the procedure, I can just cut the stitch and I know the rest will be, will pass through the GI tract, let's say, okay? In this device, would you be committed to the second anchor at all times, or is that an option?
It's a really good question. In our current prototype, that would not be an issue. In the one with a smooth suture that passes through both anchors, that certainly will not be an issue. You can pull the device outside of the skin at any point, and pulling the suture will pull through that first anchor and leave it behind. You wouldn't be able to retrieve that anchor again, but if you deploy the first one, you don't like where it is, you can back the device out, completely pull the suture out, and you're just left with your one anchor that you have deployed.
Thank you all for your questions and a very interesting device. Our next abstract in this session is entitled Contrast Enhanced Colostography, New Applications in Perioperative Evaluation of Anorectal Malformation, presented by Dr. Timothy Terrell from Boston Children's.
Hi, I'm Tim Terrell, a research resident at Boston Children's Hospital. Thanks for joining us here today. I'm going to be discussing some of the work that's been happening here to try and reframe how we think about the preoperative workup for children with anorectal malformations. We have no disclosures. Now, we know that understanding the details of anatomic relationships between pelvic structures is a critical piece of preoperative planning prior to surgical repair for children with anorectal malformations. Specifically, for imperforate anus patients, we are primarily interested in the location of the fistula between the rectum and the urinary tract, as well as the location of the rectum in relation to the coccyx, as these parameters can direct our surgical approach. Establishing the location of the fistula is considered to be the most important piece of information to be defined when planning repair of anorectal malformation. There are multiple types of imaging techniques that can be utilized to help elucidate these relationships, but involve ionizing radiation, may require sedation, or may require distension of the rectum to supraphysiologic intraluminal pressures. An optimal study would provide this information without the need for radiation or anesthesia, and also pose minimal risk to the patient. Some recent developments in the field of contrast agents have allowed the emergence of an ultrasound-based study that avoids these risks while still providing high-resolution information in 3 dimensions. Contrast-enhanced ultrasound was originally used in children to evaluate for vesicoureteral reflux and has been found to be more sensitive than fluoroscopic VCUG. Preliminary work from our institution has established feasibility of utilizing this technique to evaluate patients with anorectal malformations. So we performed a retrospective review of all the children at our institution with imperforate anus who underwent contrast-enhanced ultrasound studies and compared the results of these studies with their traditional imaging, which was usually a typical colostogram. There were 14 children with anorectal malformations and their pathologies are shown here. The colostogram for ultrasound is performed in a very similar manner to a traditional colostogram. A Foley catheter is placed in the mucous fistula, and contrast agents is instilled via gravity. There are several possible ultrasound imaging planes, and we have found the anterior sagittal plane to be the most useful. Manual pressure was used as needed in the fluoroscopic studies to distend the rectum. Our results are summarized here. Of the 14 patients, 12 of them underwent the studies as described. In 10 of the patients, contrast-enhanced ultrasound and traditional imaging yielded the same diagnostic information. 2 patients had initial ultrasound studies showing a fistula, but traditional colostogram that did not. A fistula was still not seen in one of these patients even after the application of higher pressures. And the other patient underwent a repeat colostogram, which ultimately did show the fistula. On the other side, we find that 2 patients had mucous fistulas that had scarred over. One of these patients had their fistula open in the operating room and underwent ultrasound imaging there, which successfully demonstrated the fistula. The other patient had a VCUG, which showed the fistula, but was not ultimately demonstrated on ultrasound. This is one example of a contrast-enhanced ultrasound. This image is obtained in the anterior sagittal imaging plane, so the top of the image is anterior and the bottom of the image is posterior. The bony elements of the sacrum and coccyx are seen. As contrast is instilled, the rectum can be seen filling, and then the fistula fills and contrast can be seen in the urethra and in this case refluxing into the bladder. Shown here is the traditional colostogram for comparison. In this example, you can watch the contrast fill the rectum and then see it pass through the urethra. There's no reflux into the bladder on this study, but the rectobulbar fistula is clearly defined as well as the location of the coccyx. This patient's initial colostogram was performed without high pressure and did not demonstrate a fistula. Contrast-enhanced ultrasound showed the fistula, which was confirmed on high-pressure colostogram. So what does this all mean? I think overall, our results are very promising and suggest that we should think about incorporating ultrasound into the preoperative workup for these patients. Concordant information was found in the majority of the patients, and there were some patients in whom contrast-enhanced ultrasound picked up the fistula when traditional colostogram did not. We demonstrated flexibility of this technique by showing successful fistula identification by ultrasound in the operating room. In addition, none of the ultrasound patients required additional pressure to demonstrate the fistula. While rectal damage from pressurized colostogram is rare, it has been important, so we think it's beneficial to reduce that risk as much as possible. In all, our data suggests that contrast-enhanced ultrasound is diagnostically accurate, with high sensitivity for detection of small fistulas. It's also safe, eliminating the need for ionizing radiation, sedation, and high intraluminal rectal pressures. We think that this technique is incredibly promising and are happy to share with others the experiences and lessons that we've learned along the way. If you're interested in bringing this to your institution, Dr. Dickey is the surgeon and Dr. Chow is the radiologist here at Boston Children's who have really helped make this happen. So thanks for joining us, and please reach out with any questions or comments. Thank you.
Thanks for inviting me to discuss this paper. I really enjoyed this paper a lot. We have a few questions regarding this. Ultrasound reveals most information when used as a dynamic study. What is the exact setting of your contrast ultrasound examination? How do you do it? Do the pediatric radiologist and the surgeon work as a team to optimize the study results, or is there actually a tech involved, and who does the sonography?
Well, thanks so much for your review and for your questions. So we started out with a group of a few surgeons and a small group of radiologists who were comfortable with both ultrasound and fluoroscopy. This was mostly facilitate to be able to do it at the same time as the fluorescent colostogram and to maintain reproducibility of the technique. The ultrasound was specifically performed by the radiologists in the presence of the surgeons. However, the ultrasound is portable and flexible and fairly straightforward to do once it's understood. We think there's certainly a situation where the ultrasound is performed maybe on an outpatient basis or by a technician, or perhaps in the OR just before surgery.
Dr. L, I want to congratulate you again on the studies. I really enjoyed it. I think it's a pushing envelope, but it's going to be the future, I believe, because of radiation issues with fluoroscopy. My question is, there are some centers that will still do the repair primarily on the very low rectal malformation in some selected children. So how do you utilize the sonography for this decision making? Because you can't really put a pressure to delineate where the fistula is. My second question related to the technique is, can you apply enough pressure to be able to visualize fistula with the ultrasound?
Thanks for your question. I'll take the first one in regards to the neonatal population. So we haven't yet utilized ultrasound in evaluation of neonates, but I can certainly picture a situation wherein it might be beneficial. Specifically, it's important to keep in mind that these patients would probably get something more akin to a VCUG because we don't actually have access to the distal colon. And that's certainly something that could be studied as this technique gains broader appeal. Your second question was in regards to whether or not enough pressure is applied during the ultrasound to demonstrate the fistula. And that actually is something that we didn't talk about a lot, but we think that is a benefit of this procedure, is that the ultrasound showed the fistula without needing excessively high intraluminal colonic pressure. The intraluminal colonic pressure is something that has made some radiologists uncomfortable, and there have been reports of colonic damage secondary to this, although it is rare. But in our ultrasound, we have not needed to utilize extra pressure to demonstrate the fistulas.
Your goal is obviously to avoid radiation for babies, and you have shown that the contrast-enhanced sonographic colostography isn't accurate in your hands. And have you moved on now to eliminate the fluoroscopy study?
So we haven't quite taken the final step of completely eliminating the fluoroscopic colostogram, but I will say that in the past month or so, the radiologists involved in this study have started education of this technique throughout their department in order to disseminate more broadly the technique and how it's performed. So I do anticipate that in the near future, probably at least some of the early adopting surgeons at our institution will take a step in that direction.
Thank you to the presenters and all of our invited discussants. And we will move on to the didactic sessions now. Thank you.
Hi, my name is Sean Barnett, and I've been asked by the New Technology Committee to discuss healthcare value analysis in the perioperative setting. As with any large committee or group, healthcare value analysis certainly has a mission statement. And the important parts, though, I would say, is that value analysis is an effort to collaborate, utilizing appropriate utilization, clinical efficacy, and safety issues for optimal financial value. And the key is that it's not always about the financial value that's important, but oftentimes the clinical efficacy and safety is what we really focus on. But it's not just bringing a new product to market. There's a lot that goes into value analysis. When you look at healthcare value analysis, there's really three main players that are involved in the whole process. The first, obviously, is the clinicians. Clinicians are the individuals that have the most product knowledge, and they're usually the ones that are bringing in product to the value analysis committee to try to bring it into the hospital. But you also have financial experts, which can do a cost-benefit analysis to make sure that it's reasonable to bring the product in to the healthcare setting. And you also have the supply chain and vendor management folks to actually work on the contracts and actually bring the product into the system as a whole. So together they really make up the value analysis team. When you look at a team structure, the keys though is you have a chair, which should always be the surgeon champion, and oftentimes you have a co-chair, which is going to be a nurse manager who's actually kind of charged with leading the whole value analysis process. And I would always caution you that, especially in a perioperative setting, the chair should always be a surgeon champion. Surgeon champion doesn't necessarily mean that it's the surgeon in chief, but it should be someone that has a keen interest in value analysis and the best interests of all the surgical specialties in mind. When you look at the other team members that are listed here, obviously you have biomed who would have to work on the products and help fix them, facilitate their use. Infection control, obviously, health and safety, legal to check on FDA issues and recalls. And you also have a strong education component, not only for the clinicians themselves, but for the nurses as well as the surgical techs that are actually preparing the products for us to use in the operating room. But I would say the key members are the frontline staff. And when I talk about frontline staff, I really mean those individuals that are the service leads for each specific service. And most hospitals, a pediatric surgery team will have either a nurse or a surgical scrub tech that is the service lead for them. They're the ones that are actually going to be bringing the product information to the value analysis committee meetings. They're usually the point person for all the surgeons together. Almost every specialty has that. When you look at value analysis from a methodology standpoint, there's really 5 things that happen. First is the identify stage. Then you gather data and set goals. You go through the analysis and gain approval. You acquire the product and bring it into the system itself, and then you monitor it throughout its lifespan. So let's talk about the identification first. Oftentimes we're bringing in new products into the hospital because we've identified a new problem. And sometimes it's a new product that helps with a new problem. But that's not always the case. Sometimes it's a practice change. Oftentimes we have instrumentation or devices that we use that are taken off the market, and we need a new device to supplement that use. Sometimes it's a cost-saving initiative. Tell you for high-value instrumentation and products like trocars or staplers, oftentimes you'll have trials because there is a significant cost savings that can be had depending on what the contracts there are. But I will warn you, though, that especially as a surgeon champion, you need to be really careful and make sure that everyone is on board before you start to switch just for a small amount of cost savings. Also, sometimes you're identifying that there needs to be standardization. There may be 5, 6, sometimes 7 different products that do the same thing that's in your hospital, and sometimes there's only 2 or 3 of them that are being used. And it behooves the hospital to get rid of the stuff that they're not using, not only create shelf space, but to make things easier in the buying process. And lastly, sometimes, unfortunately, products get recalled. What you'll see here is a prototypical intake form. The majority of the form isn't important. What I want you to see here, though, is that there's general themes that most of your form should have, and it should just be a standardized form to make sure that everything is the same. At first, you have basic contact information. This is generally that service lead that you have for either pediatric surgery or plastic surgery who's really bringing the product to the committee. Next, you'll see there's the manufacturer and product information. That's really just stating who the manufacturer is, what the product codes are, and everything else to make it a little bit easier to order this stuff. But the majority of the form, as you can see, is really product rationale and usage information. And it really gives committee an idea of how much it's going to be used, how it's going to be used, and why you want it. And another real key point is what it's going to replace. And lastly, there's reimbursement consideration And generally, a lot of the stuff that we're using, it's patient chargeable, so the hospital can get reimbursed for the. Next is the gather data and set goal stage. Really here you're identifying the requirements, trying to figure out how you can define clinical outcomes and how you can compare this new product to a previous product. You're looking at workflow processes, protocols, and policies that your hospital already has in place to see how those policies may be impacted by this new product that you're bringing into the hospital, because sometimes policies need to be changed. Also, as a literature review, sometimes it's pretty extensive, sometimes it's not. I can tell you oftentimes the folks that are working with you on these products that are bringing them into the hospitals have a wealth of knowledge of literature, but you usually should do a little bit of literature review on your own as well. It's also nice to know what the usage is, costs are going to be, especially from a hospital standpoint, and how it's going to be reimbursed, and what supplier capacity is to make sure that you can actually bring the stuff in the kind of numbers that you're talking about. The key, though, is you really want to identify a key clinical stakeholder in this whole process, someone that's really going to champion this through to the end and is going to be kind of the point person for people that have problems and questions, especially when they're going through the whole evaluation process. Next is analysis and approval. I can tell you there's some products that go through a pretty extensive evaluation. There's some that don't. It really just depends on the product and what it's used for. But you always need to identify a champion. You always need to have one person that's going to be that point person that's going to know the most about the product and is going to be able to ask not only the hard questions, but help with some of the easy questions that people have in regards to how it's used. You should always have an objective evaluation form. Usually these are pretty standardized, just makes it a little bit easier to compare apples to apples when you're looking at different products. But you want to have the ability to customize them depending on what you're evaluating the product for. Length of the evaluation really depends on how often it's used. There's some things that we use in pediatric surgery that we use every day. And obviously, if you're using it every day, it doesn't take very long to go through an evaluation process. But there's other things that we may only use once a week or even once a month. So the evaluation process really just needs to be tailored to product when you're deciding how long you want to do an evaluation before you feel like you really got a sense if it's going to work or not for your institution. And there certainly has to be training for those involved in the evaluation, the scrub techs as well as the clinicians, to make sure that everything goes pretty smooth. So once you complete the analysis, there'll be a cost analysis that's done as well. Oftentimes, I can tell you that the new product will end up costing a little bit more than the old product, but if there's clinical justification or safety justification, you want to make sure you have that so you can justify spending a little bit more money to make things easier in the long run. Ultimately, there'll be a contract that'll be generated, and then there'll be purchasing agreements that'll be made. Sometimes, depending on what kind of product it is, there'll either be a lease agreement or a consignment agreement. Finally, then you get into the acquisition and implementation phase. There's a lot of things going on behind the scenes during this phase. The inventory management folks are really going through the logistical plans of how we're going to get these products in, who we're going to use to actually have them there, and you really need to know what the lead time is as well. So if it's something that takes a week to get in, you really want to stay on top of that, whereas a lot of things, depending on where the warehouses are located, sometimes you can get them the same day. They usually will add them to a master inventory list so they can keep track of everything, and they'll generate a charge code so they can bill for these products as we use them. And then from the clinical side, there's a lot of education and communicating that's going on. You change policies if you need to. Oftentimes it's from an educational standpoint that you really need to change some of the policies. You decide on a go live date, and oftentimes you're going to still have existing product that's going to be in place, especially if you're replacing something with something else. So sometimes if you're using the same manufacturer, they'll exchange those one for one. Sometimes you have to use up the old product before you can actually bring the new product in, so you don't lose a lot of money off of it. And lastly, and which some people forget, but really is important, is the monitoring and sustaining portion of the value analysis. You really need to continue to monitor to make sure that your metrics are still being met, not only financially, but clinically and from a safety standpoint. Have you achieved your final goal? Is the product actually being used? Did you bring it in and just nobody decided they wanted to use it? The other key thing is that you really want to be diligent on your contracts, because these contracts are usually for 1 or 2 years, and then they revert to a different contract. And some cost savings that you had initially, you may actually find that you're paying more in the long run if someone's not keeping track of these contracts. So you really want to stay on top of that. And finally, are there any safety issues that the staff has really brought to the forefront? And if there is, you may need some re-education, or you may need to even look at a different product in the long run. So finally, as you can see, value analysis is a team approach. It's not really just bringing a new product into the hospital. There's a lot of things that go behind it. But the key is, especially from us, from a surgical standpoint, is you really need to have a surgeon champion that leads this. So you can make sure that it's not just financial based, but it's also clinically based and it's also safety based. And thank you for letting me talk. I'm happy to answer any questions that anybody may have.
Well, greetings, everyone. Whatever time zone you're joining us from today, I appreciate you taking the time to hear a little bit about the journey I've been on from the idea to the commercialization of a technology called Flextex. I'm certainly not here today to sell you Flextex, really just to tell you about my experience in medical device development and hopefully to provide some inspiration to those of you who may have an interest in such endeavors. So the medical device process, there is no like absolute roadmap, but this just highlights some of the steps. Initially, we start with an idea, something we see that maybe we're seeing a workaround that someone's doing, or how can we improve that situation? And whatever it is that inspires this seeing the need, we can then try to understand that and validate that need. Do others see that? Has anybody else tried to solve this problem before, and how have they tried? So you go through a number of those steps and eventually start to think of, what would it be that could really lead to a solution to this opportunity? And there can be a discipline to that process, and certainly involving other people in different backgrounds to help with concepts and design, help you do some prototyping. And you go down a process and try to create something that you can look at and say, okay, is this potentially going to solve the problem? Can I sell it at the right price point? I'm not defying any laws of physics or anything to make it. And if you get through those steps, you can potentially get to a point where you can put all this together and now get somebody to invest in it. That could initially be a grant. It could be outside investment, creating an entity. Whatever the path, those are the next steps to getting you onto a potential step of commercialization, which is where the outside world will invest in this. And this requires learning some new languages, understanding, potentially a manufacturing process, sales and marketing, intellectual property process, regulatory process. So there's a lot there, some new language to learn, but it's also all stuff that anybody can really understand if they dig in. And with Flextex specifically, for us, the opportunity or the need was that space between robotics and straight sticks. So providing intuitive control and articulation, but in a low-cost handheld package. That was really what we set out to do. The initial start to Flextex came from kind of a chance meeting when I was up on our engineering campus, and I met a young engineer who had just come here from MIT. And we hit it off and I started talking about my crazy ideas, and he said, that sounds cool, let's do it. So we set out, and some of the initial prototypes looked like, something out of a medieval movie, but we went through different stages. This baffle design actually showed some functionality, which was kind of a, exciting point. And we keep evolving. This particular prototype actually has a fundamental part, the virtual center mechanism, that is still in our device today. We came up with a name, Flextex, and some color schemes that started out with the device. And here you can see this advancing further. We got a big jump with National Science Foundation Small Business Awards. So these small business awards are across all the institutes. They are awesome funding mechanisms. They help you to move a technology forward, and then ultimately for you to become a commercial entity that will really take a technology and impact the world. We eventually got phase 2 funding. The key thing about phase 2 funding is that they will provide you up to $500,000 in additional funding if you raise $1 million. So we went out, we actually raised $2.4 million, opened up now a commercial space and became renters, hired our first employees, and all engineers really working to move the product forward. And again, went through a number of stages. We ultimately get to our pre-clinical device. This is ultimately, this is one of the first devices that was manufactured, and is the first device that we commercialized. So really a big milestone to hit this and, build the device. And this allowed us to get some further funding through the National Science Foundation, as well as raise some initial money. And in this quick video clip, you can see that the thing actually works. It articulates, it has this unique twirl function that allows suturing, and this was really rewarding to start from a concept to actually get to something that, you can use and functions. And we then got to the point of ready for a clinical launch, and after a lot of discussion, I did do the first case. This was super exciting, but truthfully, the joy for me has come from watching other surgeons use the device and seeing what they've been able to do with some just amazing surgeries. But our first case went great. Patient did great. So this was clearly a momentous day for our entire Flextex family and team. Our initial market experience, we launched that product in about the second quarter of 2017. We've performed over 2,000 surgeries around the world. These have been across primarily general surgery, a good number in urology, thoracic surgery, GYN, even a fair amount of pediatric surgery and pediatric urology. And through this, we have validated our core technology. And really excitingly, we've shown that we can enable surgeons to go from doing an open procedure like a Whipple or a partial nephrectomy to being able to do the procedure laparoscopically, much like the robot has. And that's been really important for us to see. That initial experience has allowed us to provide some additional funding to the company. But we've definitely hit some strong headwinds. We ran into a fact that our product cost us too much to manufacture, and that price sensitivity affected our sales and our revenue. We learned a lot about the value analysis teams or committees at all the hospitals and health systems. This is a barrier to getting a new product in, and truthfully a barrier to innovation that we all have to advocate for and monitor. We had so much excitement around our device, we went too broad with our sales. And we found that, that it takes that one-on-one effort to change surgeon behavior, which isn't always easy, and get them to commit to training. We had the normal product development delays, had some normal challenges with, management team, made some failed agreements, which all this is, sort of par for the course of any startup company. You get blindsided by some things, though. The ethylene oxide, the most common way that most medical devices are sterilized, ran into some problems, and many of the facilities were shut down, and that affected our product development and also our future product development. Fortunately, we've been able to inch along with some additional funding from our current investors, and we've made a major pivot. That's a term used when you change really your direction. So we went from single use to a multi-use reusable product, which allows us to really now bring our cost per case down into the $200 to $300 range, which is much more viable than where we were with the single use disposable product. We'll initially launch 3 needle drivers on this platform, but ultimately where we want to be and where we set out to be when we founded this company was to have a basic suite of instruments. We don't need every instrument tip, but the instruments that are beneficial to be articulating, your Maryland dissector, your hook cautery, your shears. So with some further funding, we will develop that basic suite of instruments, and this will really be a critical milestone for the future of the company. Some general challenges that any medical device company faces is you have these technical risks, right? Can you make it? How long will it take you? How much money will it take? What regulatory path are you on? Is it a high-risk Class 3 device, a Class 1 device? Recruiting talent's always, having the right people on the bus is critical, and you have to understand your competition. You have to understand the IP space and how you're going to create a protection of your product and sale around the world. And then funding, you want the money, of course, to move you forward, but how you raise the money, what terms it's raised under. So really important to try to understand those processes. You will make mistakes. We've made many. And then, execution is always critical. By that, I really mean doing things as efficiently and with the least amount of money. And it's ultimately you're trying to get to what we call an exit or a liquidity event. So how can I get to the point where somebody will now want to acquire my company, which is the most common way medical device companies exit, or that we go public. So there are other ways to get there, but those are the two main ways that a medical device company is going to get to a liquidity event. When you start a path like this, it's not going to be easy. You have to have that passion and stick to your vision. We were told by our tech transfer office this will never be a product, but we stuck to it, and you always want to go back to the need. Are you really solving the problem? You have to continue to always understand that value to price relationship, because if you're not providing value at the right price, you're not going to be successful. Finding collaborators from diverse background. Shori and I are completely different in every aspect of our backgrounds, but that's really allowed us to get a really unique relationship, and I think allows people to solve problems. And you want advice. You will seek advice from many people. You'll get some that's unsolicited. I listen to it all, but just understand that some of it is just complete BS. Don't wait for that knight in shining armor. By that I mean, we were sometimes waiting. Well, we need a CEO, or we can't do this, or we need this person, but it's not always going to be there. You have to still try to make progress, moving your project forward, and then you will attract talent as you go through those steps. Some additional thoughts when you go out and you talk to people about your technology and need money is, you kind of ask them for advice, not for the money. You say, hey, look, what do you think about this? I know our team's created. Who do you know who might be interested in this? And usually those conversations will then lead to either funding or helping you find people that might fund it. You need to develop a network, now a new network outside of your surgical network of people that can help you, mentor you, give you direction, point you to resources. And then something that didn't exist, truthfully, when we started this project is there are tons of internal grants at most institutions and also state and regional grants. And then, of course, small business grant program that I talked about. These are just phenomenal resources to help you move your project forward. Of course, they're competitive, but that also helps you to have that discipline and really think what you're doing. And then, one challenge is the finish line always keeps moving, right? So first looks like 100 yards, and then, you get almost to the tape, ready to put your arms up, and then, it's around the track another time. So that's just normal. You have to embrace it, enjoy it, and remember it's not about, that end. It's not about just crossing that finish line, but it's about the journey. There's amazing depth of relationships through this process, amazing depth of experience, and so much joy. I will tell you, there's definitely been tears, but I have really enjoyed it, had tons of fun. And if you stay on it, and if you, I think for all of us, truthfully, it's about creating something. And it doesn't have to be a medical device. It could be a software. It could be something you write. It's something that somebody else can consume, right? And even better if they pay for it or repeatedly pay for it so that you can make more for the world. But whatever the model is that moves it forward, if you can create something, you can impact even more lives than we do in the great blessings that we have each day to impact a single life through an operation. And anyway, I look forward to seeing your guys' progress. I hope maybe I provided just a little bit of inspiration to get some of you to maybe take this path. And certainly, if I can be of help in any way, you don't have to listen, but I'd be happy to provide you any advice. So anyway, thank you very much for your attention, and God bless.